Glad to see the room completely packed for one of our candidates,
The free lunch at your house.
Robert Hill, who's being interviewed for a faculty position in the Center for Glial Biology at the VTCRI. Robert grew up in a farm, I think, in Montana, is that correct? And escaped that country life by going to Hartford to Trinity College for his undergraduate work, small liberal arts college there, and got a BS in neuroscience. And after getting the neuroscience bug, went into a graduate program at UConn's, just a stone throw away, where he worked with Kiko Nikiyama, who at that time had just developed a really important tool for glial biologists, a transgenic animal in which we can identify NG2 glial progenitor cells that we have since learned are the progenitor cells for oligodendrocytes. And really his work since has been focused on studying these NG2 cells. During his graduate work, he looked at lineage relationships, what all can form from these NG2 cells. He found that in certain regions of the brain they can give rise to astrocytes. He looked at development of these cells and found that there is a time window postnatally, somewhere around 5 to 8 days of age, where they were differentiating into oligodendrocytes. And if you do sensory deprivation, you can actually open that time window. So they have a delay in terms of their differentiation and instead continue to proliferate. He's published several really high-impact papers, including a Nature Neuroscience paper during his graduate work. He then went for his postdoc work to Yale to work with Jamie Gutzendler, who is really a pioneer in using multiphoton imaging in studying glial cells, vascular cells, neurons in vivo. And there is where he got involved in developing really a whole set of new tools to look at oligodendrocytes and myelin in specific. As you see on this image, he's going to explain to us, this is a confocal reflection microscopy image where you can, without putting any additional label or fluorescent protein in there, by spectroscopic methods using different wavelengths of laser light, image myelin and image myelin in a dynamic fashion, look at changes in myelination in vivo. And he's going to tell us a lot about that today. He's published 11 papers in very high impact papers, Nature Neuroscience, Nature Methods, Nature Biotechnology, Neuron, Journal of Neuroscience. He has several more in various stages of preparation. He's been able to support himself throughout undergraduate and graduate work. He had an NRSA, and he's successfully completed for a K99 award as well. So it's really a pleasure to have him here to talk about uncovering mechanisms of myelin plasticity in the living brain.
Okay, so I'd like to thank Dr. Songheimer for the kind introduction, and also the search committee for inviting me. I'm very excited to be here. I've had a great time so far and looking forward to meeting more people throughout today and tomorrow. So as Dr. Songheimer said, I'm interested in myelin and how it might change in the adult brain. And what you're looking at here is in the cyan you see myelinated fibers that are imaged through a new technique that we have developed, and then in red we have the cortical blood vessels, and this is captured from the a live mouse brain. You're basically looking down onto the cortex of a live mouse, and I'll tell you about how we generate this type of image, and then what we can use it for, and how it allows us to ask a whole new set of questions about myelin plasticity in the live brain. So first I want to give a real brief introduction about what my approach is and how I would want to move forward with this same type of approach. And basically, as I mentioned, I'm interested in myelin formation, plasticity, and maintenance. And how I go about this is by developing new in vivo optical imaging and manipulation approaches. I think tool development is central to our advancement in understanding how our bodies work. And I think once you develop a new tool, you're able to address new questions that no one else has been able to do before. It puts you in a very unique and powerful position, and I think it's one of the best ways to advance our science. And so, but then you also have to use those new tools to understand, start applying those tools. So I want to have a balance between developing these new tools and then applying them, specifically primary focus on myelin formation, but also interactions between multiple cell types in the brain and neuron-glia interactions in the live brain. So today I'll tell you about three different kind of general topics. The first one would be a project that I started in graduate school looking at the mechanisms of oligodendrocyte generation, so looking at the differentiation from the progenitor cell into the mature oligodendrocyte. The next thing I'll talk about is a toolbox that we have developed for in vivo optical imaging of these cell types. And then the last thing I'll talk about is using this new toolbox for some investigation of lifelong myelin plasticity in the brain. So I'll give a brief introduction about myelination, just so we're all on the same page. It's basically the insulation for your axons, allows rapid propagation of an action potential down an axon, and it increases the conduction efficiency while also increasing the speed, and it's been shown to provide some trophic support for these long axons. And so this is basically the schematic that you hear or you see when you learn about myelination in a basic introductory course. And while this is obviously very important, I think this is a very way oversimplified, and it's a little bit boring. And hopefully I'll be able to convince you that it's more than just insulation, and that this myelination could be playing a much more active role, and it's much more dynamic in the adult brain. And I think our understanding of this process is very rudimentary at this point. So hopefully you'll be convinced and more excited about myelination, that it's not just insulation. So another interesting thing about myelin is that it's primarily been studied in white matter regions for good reasons in the brain. So what you're looking at here is a coronal section taken from the mouse brain. This is one hemisphere, and you have a white matter region here. Myelin is stained by myelin basic protein, and it's a dark color here. And it's primarily been studied in these white matter regions for good reason, because it's concentrated in those regions. You have bundles of axons traveling through these regions, and a lot of them are heavily myelinated. But in addition to that, in cerebral cortex or other gray matter regions, you also have a considerable amount of myelination, and it's very much understudied, and I think it could have very important roles in cortical circuitry. And another good reason is that it's accessible for in vivo optical imaging techniques. And so here's just an example of, if you zoom in on the cerebral cortex, you can see this is the peel surface in a mouse, and we're stained for another myelin-specific protein, CMPase. And what's very interesting is in layer 1, you have this big band of myelinated fibers. And this is because there's a bunch of axons traversing across your hemisphere, or from one hemisphere to the next, or even within the same hemisphere. And a lot of these axons are myelinated. So this is a very interesting, kind of blanket of axons that are accessible for optical imaging, and also we know very little about them. So it's kind of ideal situation for studying these in the live mouse. And so myelination is made by oligodendrocytes in the central nervous system. Here's an example of a single oligodendrocyte that's labeled in a transgenic mouse with membrane-bound fluorescent protein. So the cell body would be here, and then each one of these processes we call an internode. And each one of these processes, you can have up to 60 from a single oligodendrocyte, contacts up to 60 different axons. Each internode here will be contacting a different axon. And if you take the cross-section of one of these internodes and look at this at the ultrastructural level, you can see what the myelination is. It's basically a multi-layered membrane extension from this oligodendrocyte wrapping around a single axon. And this multi-layered compact membrane allows for this rapid conduction and changes the efficiency of the action potential conduction down the axon. And so another very interesting thing about these cells is that they're the only cell in the cerebral cortex that is continuously generated even into adulthood. And this is possible due to this progenitor population that I'll be calling NG2 glia here. They have a bunch of different names, but I'll refer to them as NG2 glia. And they're basically considered now the fourth major glial cell type in the brain. This shows you how abundant they are. This is a transgenic mouse that has specific labeling of these cell types. They're tiled throughout both gray and white matter regions. Here, this is the cerebral cortex. This is the corpus callosum. So you can see they're quite dense. They're almost as dense as something like a microglia. And they're highly proliferative, even in the adult brain, and they continuously generate oligodendrocytes. So here's just a good example of what their morphology is, and here's an example of even a pair of cells that we had caught during the division process. So we have a bunch of different transgenic mouse lines now that we can image these cells, and here's an example of how proliferative these cells are and how much they move around in the live brain. So this is a mouse. It's the postnatal age is indicated here. You image these cells every 2 days using a 2-photon microscope and looking, do an image down into the brain. And you can see here's a cell at postnatal day 50 that divides once. Oh, sorry. Well, anyway, I think you probably saw that, but I don't know why it stopped working, but anyway, it divides once, and then you can see that it divides again, and where there's other cells here, such as pericytes, that are labeled in this mouse, but basically we have a way to look at the dynamics of these cells, and you can see how dynamic in a mouse that would be considered adult for a lot of developmental processes, but even though these cells are continuously proliferating and generating oligodendrocytes. So we have this interesting system where we have a resident progenitor population. They proliferate, they differentiate into oligodendrocytes. Sometimes they go through programmed cell death through apoptosis. They generate this oligodendrocyte, which has different markers, has a completely different morphology. And then once it's a oligodendrocyte, and we know a lot about this process, at least in culture, a lot of molecular signals that influence this differentiation process. A lot of knockout animals will have defects in this differentiation process. But once you get to the oligodendrocyte, we know very little about their dynamics, such as how stable a single oligodendrocyte is, whether or not how much they're turned over, whether or not they have any structural plasticity. So once they're what we would normally call a mature oligodendrocyte forming compact myelin at each internode, whether or not these structures can change, we don't know. We know very little about that in the live brain. And then also, once you generate a new oligodendrocyte, how it incorporates into the neural network. So basically, how it decides that this axon is going to become myelinated while this one's going to remain unmyelinated. Whether or not that's a signal from the oligodendrocyte, a signal from the axon, or how these all work together, there's still a lot of unknowns about that. And then this potentially, as these new cells get incorporated in adulthood, these have the potential to then modulate the action potential propagation and potential circuit dynamics. Because as you can imagine, if you all of a sudden have one of the axon in the circuit that's myelinated, it changes the conduction down that axon, and the timing will change based on just that addition of the new myelination. And since you have this progenitor population that is maintained, these changes in the circuit or changes in, say, activity of neurons in different circuits could potentially then feed back on this process, and you could generate more cells or change the dynamics of this whole process. So I think it's a very interesting system that normally you would only see this type of thing during development, but for these cell types it seems to be important and it's maintained in the adult. So I'm very interested in studying all these processes, and then once you understand how this process works in the normal situation, you can look at the signals that influence it, the developmental progression, and then how it's disrupted in diseases. And so to give you a kind of elegant example of how myelination along a single axon could be very important for how a circuit works. There's a very nice system in the chicken auditory brainstem that's been shown. If you have an auditory signal coming in to a single neuron here, and that will have an axon coming out, and this is from the same axon. You have one branch going to the ipsilateral side, one branch going to the contralateral side. And if you look at those branches, the myelination on each axon is different. This one has to travel twice the distance that this one does. So the myelination actually gets set up that you have the internodes that are much longer. And the reason that this is important is because for coincidence detection and sound localization in this system, you need the signal to arrive here at the same time that it arrives here. So this is a very interesting I think an elegant example of how myelination specifically can influence a circuit and how it works. And this type of thing is likely happening in a lot of different brain regions. It's just we don't have such a clean way of having a readout for it. So I think it makes it very important for us to learn more about it. And so it's likely that a lot of these you can imagine you lose a couple of these internodes along this axon, it could have dramatic influences in disease, and such as demyelinating disorders like multiple sclerosis, but also in other conditions such as aging, Alzheimer's disease, and schizophrenia. It's been shown in fixed tissue that in these conditions, myelination has degenerated. So the first thing I'm going to talk about is a quick study about how we analyzed this decision to differentiate and whether or not this decision is connected to cell division and modulated by external signals. And this was started in graduate school, and I continued a little bit in my postdoctoral fellowship, but it's primarily during my graduate school work. And so I started this by doing a fairly simple experiment. We have these NG2 glia, oligodendrocyte progenitor cells. We have a transgenic mice that we can tag them when they're at this stage, and tag them with a YFP reporter by injecting tamoxifen. And then, since we know that at that stage that we've injected the tamoxifen, they're all the progenitor stage, we could then do a simple fate mapping experiment to determine when these cells differentiate into mature oligodendrocytes. And on top of that, we can also do a pulse chase experiment with the EDU, which will be incorporated when the cells are undergoing cell division. And so we can combine this fate mapping with a pulse chase and then determine how long it takes these cells to differentiate into oligodendrocyte in vivo. So this is a very straightforward question, how long this differentiation process takes and whether or not we can somehow connect it to cell division. So this was primarily done during early postnatal development. And basically, when you do this experiment, you can have a couple different outcomes of what if you analyze single cell pairs. So basically, if you look one day after the pulse of EDU, you can see pairs of cells that have maintained at the progenitor stage. If you look 3 days later, you can also see pairs of cells that have maintained at the progenitor stage. But you can also see other examples where one cell that's from this pair had differentiated into oligodendrocyte by this marker, while the other cell did not, or then both cells differentiated into oligodendrocyte. So basically, we can see all the outcomes that you might expect, the fate outcomes, either one or both of the cells differentiating into oligodendrocyte or staying the same in both cortex and corpus callosum. And what was interesting about this was that we saw that if you look at within 2 to 4 days at P8 or 2 to 6 days at P21, you could see this leveling off of the proportion of cells that had differentiated into oligodendrocyte. And this suggested that the majority of the cells that had divided and then differentiated did so within this temporal window. And this seemed to be highly reliable, and it made us wonder if during this time there was, if it was potentially connected to division, but also if this is a way that we could modulate this process and see if you change something in the environment, it could potentially alter, shift this from, say, fewer of the cells differentiating or more of the cells differentiating, specifically connected to the division. And it's allowed us one of the first ways to test this type of thing in vivo. So we went through we applied a fairly simple method to manipulate the environment by sensory depriving one side of the whisker barrel cortex. So basically, we take a mouse, clip all the whiskers on one side, and then we can look in this cortex, specifically in the sides that either receive input from one whisker pad or the other, and then we have a direct internal control, and we can then do this pulse chase fate mapping experiment and see if there's any effect by depriving the sensory input on this differentiation process after division. And what we saw was that basically you can see, if you look in the deprived side, you can see fewer oligodendrocytes are generated in general, fewer of the there's also fewer fate-mapped oligodendrocytes that are generated. So basically the cells that had differentiated, this is not yet connected to division, but differentiated from the progenitor cell. We can see fewer number on the deprived side. And then also connected to division, we can see that there's a fewer oligodendrocytes that had been labeled and divided at 2 days earlier that had also differentiated. So this is an interesting system that we were able to see that sensory input was modulating this process, so potentially normal activity was influencing the production of oligodendrocytes. One of the first, one of the few papers that have shown that in vivo. And we also want to look at what's happening to these cells that are not differentiating. Are they going through some other fate? And if we did some staining for caspase, which is a marker for apoptotic cells, we actually started to see a lot of these YFP positive cells that were caspase positive, or even YFP and EDU, so cells that were NG2 glia at the time of injection, and then they had started to differentiate or divide, but then they had undergone apoptosis instead. Here's just an example of a couple more of those cells. And they sometimes faintly expressed the progenitor marker or the mature oligodendrocyte marker, but a lot of them had actually were within that lineage. And so we see a larger amount of apoptotic cells on the deprived side, and also fate mapped, and the ones that had divided. So it seems that we've discovered this that oligodendrocyte differentiation occurs during a precise window after division, at least during these developmental stages. And then sensory deprivation reduces the generation of oligodendrocytes and also increases apoptosis of divided cells. So it seems like this has the potential for modulation by external signals. Yep.
Did you do the flip experiment? That is, instead of deprivation with the whisker trimming, did you do anything with animals with whisking behavior to sort of hyperactivate and see if you could drive the apoptosis even further, for example?
Yeah, so I think it's very difficult. You can put the animals in, say, like an enriched environment or something like that to increase the sensory input, but at least for this age of animals, that may not do too much because they're P8, they're just barely walking around. But we didn't. The flip side of this that we did in this paper was we did a demyelination model in a slice culture and then looked at the division, and that actually sped up. I didn't put it in because I didn't want to spend too much time on this, but that was our flip side of it as opposed to increase in sensory input. So these experiments suggested that there's modulation by external signals, and it makes us now wonder if there's a very interesting synaptic communication between neurons and NG2 glia that occurs. So it's potential that this synaptic communication that is altered when you change the sensory input could potentially be the reason for this change in the fate from differentiating into oligodendrocyte to instead going through under apoptosis, but it's also possible that there could be other signals besides this specific synaptic input, such as growth factors that change when you change this broad, it causes a decrease in sensory input. It's not necessarily only the neurons that are changing, astrocytes, other cell types are changing when you do this, so it's not as specific. So I think there's a lot of, more experiments we could design to attest what's happening, what's controlling this change. So another thing, I'll move on to the next project now, but another thing that I hadn't mentioned yet was that during this differentiation process, it's very interesting that there's formation of gap junctions between astrocytes and oligodendrocytes. And so in addition to all the, the molecular morphological changes that occur, they somehow start forming gap junctions with oligodendrocytes. And these gap junctions seem to be very important for the maintenance and potentially the differentiation of these cells, because if you have mutations in these specific gap junctions, you can have demyelinating or hypomyelinating diseases that are quite devastating. And these gap junctions can be found both on the cell bodies between oligodendrocytes and astrocytes, and the astrocytes and the myelin sheath itself. So this is a very interesting topic that we don't know too much about, besides that there's these mutations and that they occur, and people have filled cells in slices and seen coupling between these cells, and can stain and see that these gap junctions are there. But besides that, that's mainly what we know. And as I was doing a set of experiments where I was imaging astrocytes using this dye called sulforodamine 101. I wasn't trying to do any myelin experiments here. I was, exploring and labeling other cell types and doing some other imaging experiments and using this dye. And you can see I was using this because you get a very, very nice label of the astrocytes. So that was a video showing you a 2-photon Z-stack going into the brain, where you can see individual cells that are labeled. And if you then label the vasculature with an intravascular injection of fluorescent dye, you can see a nice astrocytic infeed on the blood vessels. You can get very nice morphology. So this is a very powerful tool for labeling astrocytes. You know, a lot of people that do in vivo imaging, this is one of the first things you do. You just apply the dye to the surface of the cortex, and you can get beautiful images of the astrocytes. But when I was using this, I happened to be doing it in a mouse that had astrocytes labeled with GFP. So I guess I was trying to get a dual label of astrocytes. And basically what I saw was that cells popped up. This should be labeling all the astrocytes. Cells popped up that were not labeled with the GFP. And in converse, if you do it in a mouse that has oligodendrocyte lineage labeled, both the mature oligodendrocytes and the progenitors, you can see cells that are labeled with the sulforamine 101 and the GFP, but also cells that are not. And when I looked at these cells in a little higher resolution, you can see here's an example of two cells that are not labeled, presumed astrocytes, because this is a oligodendrocyte mouse, both the progenitor cells and the mature oligodendrocytes. But then we have single cells that are labeled with the sulforamine, and then single cells that are not. And these cells have the morphology that looks much more like the progenitor cell, while these ones look much more like the differentiated cell. And if you quantify these cells in this mouse, you can see that the proportion of these sulforamine labeled cells is quite high that are actually these oligodendrocytes. And so this is important for people using this diet. It could be, depending on the age of the animal, there could be a big proportion of the cells that you're looking at are astrocytes. But more interestingly for me, so people need to be aware of that, but more interesting for me is that what we think is happening is that the sulforamine dye gets taken up by the astrocytes, and then since they're only gap junction coupled with the mature oligodendrocytes, then the dye gets spread into the oligodendrocytes. And this potentially is a way that we could then watch when these gap junctions have formed in vivo. And so I did a preliminary experiment trying to see if that is possible. So what you're looking at here is a time-lapse image. Again, we have the postnatal day here, and basically I have oligodendrocytes labeled, the oligodendrocyte lineage, both the progenitor cells and the mature oligodendrocytes labeled with GFP, and then I repeatedly inject this sulforamine dye and at each imaging time point. And basically there's one cell here that was labeled with sulforamine throughout the whole experiment. So we would call this a mature oligodendrocyte. There's one cell here that was not labeled the first day, but then it becomes labeled. I'm not sure why these keep stopping, but. Well, I'll move on to the next one. Anyway, so this is the same time-lapse images, just showing you the crucial points when there's something that changed. But basically, we have another cell here that was not labeled with sulforamine at P50. 2 days later, it's all of a sudden labeled with the sulforamine. And then we have the third cell here that is not labeled until from P68 to P70. I think this cell has differentiated and now has started to become gap junction coupled with the astrocytes. Yep.
Is there any evidence that the nature of the connexins involved affects the kinetics of the time to the dye coupling that you see that forms the gap junction?
We haven't done any of those experiments. I think it would be good to have, you do a knockout of specific net connexins and see if it changes. The dye coupling, when you look at the reason I think that it's gap junction coupling between the cells is because you can do gap junction blockers and you inhibit this labeling. We have that in the paper. And then also if you look at the timing of the labeling on a shorter time scale where you apply the dye, then you start imaging, you see the astrocytes get labeled first actually, and then you see the oligodendrocytes. But what gap junctions specifically are necessary for that, it still needs to be worked out.
Have you thought about photo-bleaching one of the coupled cells and looking at the kinetics of it?
Yeah, we could definitely try that, yeah, especially if you have, the dye is so bright and seems very stable, but you could try that, yeah.
Octanol or something like that, or what do you use to block?
Carbonoxolone. Yeah. Okay, so that was a kind of a quick aside, but I wanted to mention that because I think it's a very interesting technique. And basically, now it's the only method that we have for detection of astrocyte gap junction coupling in vivo. And this is potentially very important because it's now we could study this process when you demyelinate a cell and it goes through remyelination. Also under situations such as neuroinflammation, it's been shown that gap junction coupling or the connexin channels can be altered, and function can be disrupted. So now we have a method for specifically testing when the gap junction coupling is altered and it might give us a hint about the roles that this coupling might play in the maintenance of these oligodendrocytes and have a direct connection to some human diseases. So I think this is a very interesting kind of a way that another technique we have for dissecting this process from the progenitor cell to the mature oligodendrocyte. So thus far, all I've been talking about is focusing on this differentiation process. But as I mentioned, there's a lot here that we don't know about the stability, turnover, the plasticity of these cells, and then how they incorporate into the network. And so I'll spend the rest part mainly focusing on that. And one reason we don't know much about this is that the imaging approaches that we've used in the past, a lot of fluorescent labeling, is not ideal for oligodendrocytes, especially for compact myelin. If you don't have membrane-bound fluorescent protein, a lot of times the cytoplasmic labeling of fluorescent protein won't get into these compact myelin-forming internodes because there's very little cytoplasm or no cytoplasm in that. So you obviously often can't even visualize the internodes. So that's one reason we haven't imaged this process, and also just the transgenic mice were not available at the time. But as we were, so we've kind of by accident, as another accident that happened in the lab, we had a postdoc that I was working with was using, instead of fluorescence imaging like I've been talking about, where you excite a fluorophore and collect the emission, he was instead doing reflection microscopy. And basically that's very simple. All it is you send down say one wavelength of laser, such as 488 nanometers, and then you collect 488, so just reflection of the same wavelength instead of fluorescence. And you can do this on most confocal microscopes now. When he was trying this in the mouse cortex in vivo, and these fibers popped out of him. So here's the example of one of the fibers that was labeled with one wavelength with 488 nanometers, and you could see you could imagine that there's a fiber here. It's a little bit patchy though, and what he's noticed was that if you do several different wavelengths, you can get similar patchiness. The same fibers are revealed, but if you combine those into one channel, you actually get that the different parts of that fiber reflect different wavelengths. And basically what this will come down to is that I'm using, for any of these images that I'm doing this score microscopy, I'm using 3 lasers and combining them into one channel, just so we don't have too many colors going on, since I combine it with the fluorescence imaging. But it's very interesting that you have these little spots that are highly reflective of one wavelength and not the next, and I'll talk a little bit about what we think this might be. But basically for most of the images, you'll see I'm combining them into one channel. And so based on the density of these fibers and just thinking about what might be highly reflective in the brain, we guess that this could be myelinated fibers, or the myelin that is highly reflective, based on its multi-layered membrane that's wrapping around the axons, and it's pretty much fat in an aqueous medium compared to the, much more reflective compared to the rest of the brain. But we had to test this in vivo, and so there was currently not really any ways to label myelin in vivo, but we tried this fluorescent dye that's called fluoromyelin. It's been used in tissue cultures and fixed tissue, and we found that if you put it on the surface of the brain, you can get very nice labeling of the myelin. You get this dual labeling of the myelin sheath, and if you combine it then with the reflection technique, you can see they actually 100% co-localize, even along single fibers. If there'll be break, there's no fluoromyelin dye, there's no score signal there either. So it seemed to be very specific to the myelinated fibers, and if you do it in a transgenic mouse that has some axons labeled, you can even see at locations such as the axonal bifurcation, where we know that there's a break in the myelination here. A lot of times there's a node of Ranvier there. We see no fluoromyelin dye labeling, and also our score technique also is not reflective right there. So it seems to be very specific to where the myelination is. And you can also use this in fixed tissue. And here's an example of the perinodal regions, the regions at the end of a single oligodendrocyte internode labeled with this Casper antibody. And then you can see here's the internode, and then there'd be a node of Ranvier, and then the next one's starting. So it seems to be very specific. This label-free technique, you don't need any, you can do it in any tissue, any animal, and it basically is, you have a label for compact myelin. So I mentioned about the colors. So you can see that you can get these very nice images of different spots along these fibers that are highly reflective for different wavelengths. And the reason we think, or what we think is going on here, is some thin film interference type mechanism. So basically what you see here is just an oil droplet on the pavement that you'll see this every time it rains. And what happens here is that you have oil that's getting thinner and changing its thickness in the water, and then the sunlight will reflect down on that, and it will have destructive or constructive interference and reflect a specific wavelength more predominantly. And so we think of something very similar to this is happening with this technique, where you have tiny nanometer changes in the myelination thickness. It's not like it's a whole wrap change. It could be just some little alteration. It's a biological structure, so it's not perfect. And tiny little changes can cause one wavelength to be reflective more than the other. And we thought one way to test this was to do this same score imaging technique. You do this imaging on a pulled glass pipette. So basically it's a glass that's highly reflective, just like the myelination is. You pull it, it gets to about the diameter of an axon, but since it's pulled, it's getting smaller and smaller. And then you do this score imaging technique, and what we can actually see is that it cycles through the different colors. These are very small changes in the diameter, but it's showing that this type of idea. We're not exactly sure this is what's going on, but this is at least our hypothesis of how this multicolor reflection, where it's coming from. And so all the images I've been showing so far are from the cortex, and you can see the multicolor is maybe its potential is useful for something, but we don't know much about it yet. But if you go into regions that have much more heterogeneous axon diameter thicknesses, such as the spinal cord or sciatic nerve, you can actually see single fibers along single axons will reflect one color more than the other. And so example of these two axons that were imaged in the sciatic nerve in vivo, one seems to reflect green way more than the other one that's reflecting the 633 laser. So it's you get these very nice, beautiful images, but you could potentially use this for tracing or something. But anyway, it's a nice way to use this score technique, and you can use the multicolor approach. So basically, we have this technique now that we can image the myelination in a label-free manner. So one of the first things you would want to do is then look at the dynamics of these fibers in vivo. And since it's a label-free technique, we can do it in very old animals, or any transgenic animal that you have, you can do this imaging right away. So basically, it's been starting to do some characterize myelination in layer 1 of the cortex at different ages. Here we have 30-day-old animals, and we have 910-day-old animals, and you can do some quite quantitative analysis of the density of these fibers, how intense the signal is, and see how prolonged the formation of new myelination is. And basically what we're finding here is that up to 640 days old in a mouse is 2 years old. It's quite into late adulthood. Some of these mice are also almost starting to look as aged mice. You can still see this quite dramatic increase in the density of these reflective fibers. So it seems that myelination is progressing linearly, almost linearly, all the way from P30 to the 640 days. And then you also have this interesting drop where you can start to see some pathology that's associated with aging. And it's kind of an interesting stat that came out of this. If you just extrapolate how much myelin is being added by length per day in 1 cubic millimeter, you see that 24 millimeters, this is a mouse layer 1 cortex, you can see that 24 millimeters of myelin by length is being added in 1 cubic millimeter per day in each, from P30 to P640, which is quite, when you think about that, it's pretty amazing that there's this much happening, pretty much throughout the life of the mouse. Yeah. Yeah. So this is, yeah, it's all layer 1. It's all within the first 50 to 75 microns. So it's not extrapolated into the whole cortex. It's like you could say the layer 1 millimeter cubed, however that would work. So those were all static images. But you can also then do repeated imaging, time-lapse imaging of this process, and look at the addition of these fibers. So this is an example of a mouse image that when it's 50 days old and 110, and you can see here's the same fibers, but there's also, so much more that's been added. So that means that there's, tons of new oligodendrocytes that were generated. All these fibers are new between these time points. And as I mentioned, this is, a lot of times this is, P50, P60 is considered adult for a lot of experiments. So it's quite dramatic how much change there is.
So you see, just quick aside, can you go back one?
Yep.
If you had a bunch of times in between there and after, if you had a whole series, like those ones you showed us, do you see for a given one the myelin coming and going, or is it just a progressive addition that we're seeing here?
Okay, yeah, so I'll get to that, the next one, yeah. So we can also, look at this in a little more detail about, we can label the oligodendrocytes and see if the formation of new fibers is due to the addition of a new oligodendrocyte, or if it's a oligodendrocyte that was already there that's then forming a new internode. And basically what we've seen is that every time you have formation of new fibers, that at least a whole fiber is due to the addition of a new cell. And so here's an example of, we have oligodendrocytes labeled with this DS-red fluorescent protein. There's a new cell here, and we see some new fibers that are labeled. This fibers just show you the same one. But in addition to that, whole new fibers being labeled, we're also observing these distinct changes in single internode processes, where we can see extension happening, where this situation, the score signal is 14 microns long, just going from there to there, and it's extended quite a bit over these 30 days. You can also see the opposite, where some of these processes are retracting. And so you can also see other interesting thing where two processes seem to be coming together over quite long periods of time. 30 days, they haven't moved too much, but they are getting closer and closer. And here's 5 days, the processes there have become quite a bit closer. So it seems like there's dynamic changes, but they're also quite prolonged. And so it's a little bit difficult to understand about what type of signals could be inducing these day-long or week-long changes in these processes. But I think it's very, very interesting that we see this type of thing. So in addition to that, you can see a lot of stability, obviously, too, where this same process is imaged over, quite a long period of time. Not much has changed here besides the addition of some new processes. But we also see some shifts in what we see the presumed location of nodes of Ranvier, which I think this is very interesting about. It must mean that one internode is getting shorter while the other one is lengthening, and what type of process or how this would change the function or the conduction of the action potential is very, very interesting. So we also wanted to, so we're using all this for the label-free technique, but wanted to confirm that what we're seeing is also, we can see it with more traditional fluorescent type labeling. So we can use other transgenic mice that have membrane-bound GFP, so we can visualize the internodes and basically image single cells in this situation. Since it's tamoxifen inducible, I can titrate the tamoxifen, label a single cell, follow that cell over time, and they're all at the mature oligodendrocyte stage, and then look at single internodes. And we see the same type of thing. There's a lot of processes that are stable. Some extend, some retract, some extend, quite long distances. And you can, quantify those. Here's examples of single cells. Have some internodes. It's not like all the internodes are getting longer on that single cell. Some internodes are getting longer, some are quite stable. This cell had some internodes getting longer, some internodes getting shorter. And here's just the distribution of those different types of dynamics. So everything I've been talking about so far is a perspective of the oligodendrocyte, but we can also take the perspective of the axon. And so basically, if along a single axon, there has to be multiple oligodendrocytes working together in order to myelinate that axon. And so if we take that perspective, we can also look at these dynamics and see how this types of distribution of myelination along single axons changes over time. And so it was recently revealed in P60 mice that if you look along single axons, you can see quite heterogeneous myelination along single axons. So here's an example of a region of the axon that has a myelination on it, and then you can see a fairly big break, and then you can see myelination start again. And these breaks go against what we normally thought is either axon being myelinated or unmyelinated. And so it's very interesting about whether or not these get filled in over time and how dynamic this situation is. So we thought we could look at this process in vivo. So basically, I've generated a way to label single axons with GFP. We can trace where these axons have a myelination along them. So here's an example of one that has a couple internodes, and here's a what presumed node of Ranvier, and then we have also regions of that that are not myelinated. You can trace these over fairly long distances in your craniotomy, in the cortex, and then you can do this at a bunch of different ages, get quantification of how much of these axons are covered by myelination. So we see this, again, this shift from 30 days old to 640 days old, where at 640 days, the vast majority of these axons are almost completely covered by myelination. There's still some very interesting that are not, though. They have big gaps in them. You have a single single internode along an axon, but there's again seems that there's very prolonged, lifelong change in the myelination of a single axon. And then you can do repeated imaging of this, where we have single axons labeled. You look at it at one age and then the next, and we can actually see axons that have only one or two internodes on them at one point, and then we can see addition of other internodes on that, such as this, where we have these internodes here, and we can see there seems to be quite a prolonged addition of new myelin on this axon. So I think this is quite powerful approach to start looking at these dynamics from the perspective of the axon. So basically what we have, the summary from this part is that we have a novel platform for in vivo studies of myelin plasticity. We see these lifelong changes in the density and the distribution along single axons. There's continual oligodendrocyte production, and there's also this long-term internode plasticity, which I think is quite interesting. So to summarize what I've talked about so far, basically we have found out a little bit about this cycle, but I think there's so much that we don't know. We see that external signals can influence the production of oligodendrocytes, such as sensory input. We also have a new method for looking at this gap junction coupling between these cells, and potentially look at that in all kinds of situations. We've observed some internode plasticity. We've also seen how these cells might start to integrate onto, if you take the perspective of the axon. We don't know much about how this might change the modulation of action potential propagation in the circuit. I think that's that's one of my future plans. So I'll go very briefly into what I would want to immediately start studying in my lab. The first one is look at a lot more of these details about how this oligodendrocyte axon unit is formed and maintained in vivo. So basically, we can now use this very broad toolbox that's all in vivo. We have all these ways to label all these different processes and image it dynamically over time, and we can look at this differentiation process, look at when this gap junction coupling occurs, see how it's correlated with the formation of compact myelin. Then we can start to label the nodes of Ranvier using some viral approaches and overexpression or CRISPR knock-in type approaches to specifically label single molecules in the axon and see how it changes when the oligodendrocyte process is changing, and then also look at this distribution along single axons. And I think things such as this internode location moving would be quite interesting to say if we see the sodium channels that are clustered at a node of Ranvier, if they move from one location to the next when you have this type of thing changing. So then you could also start to manipulate the axon, manipulate the oligodendrocyte, manipulate the environment, and you could manipulate one axon in your field of view, see how that changes, all of these processes. And we have ways to start, manipulating single axons now. And then also you can start looking at how this is disrupted in disease, and I've started to look a little bit made a way to demyelinate by ablating a single oligodendrocyte. So here's an example. I've ablated this oligodendrocyte, and then you can see that there's a little patch of demyelination. So we can then study the remyelination process. We can also look at this in aging. So I mentioned at the beginning that there's this decline in aging, and since we have this label-free technique, I've imaged mice that are 1,000 days old, and then you look a couple weeks later, and I can see some very interesting pathology occurring. You see some loss of myelination. You can see some formation of these very interesting balls of myelination that are in microglia. And then we've also made a technique for ablating cells or inducing apoptosis of cells, in this example of a neuron they were killing, but you could imagine killing a oligodendrocyte, and we have neurons labeled there, microglia labeled there, and what you're seeing is the microglia starting to phagocytose that cell, and then there's, on the bottom here is myelination within a oligodendrocyte. So I think we have, once you start looking at these multiple cell types and looking at these dynamics, that you can start to reveal a lot about how this works in vivo. And so the last thing I want to do is kind of a goal, and we're not really there yet, but I think it's a very interesting approach that I want to develop in my own lab, is basically once you have all these changes in the myelination, how does the function of the axon change? And I want to do this by using some optical approaches for looking at axon function. So here's an example of some axons that are labeled with genetically encoded calcium indicator. Ideally, you'd want to do this with a genetically encoded voltage indicator to look at changes of how the action potential conducts along an axon. But I think the voltage indicators, at least currently, may not be bright enough for some of these approaches. But basically, lots of labs are actively generating those now. So I want to use this type of approach to then look at these changes and then potentially look at an axon before and after these changes have occurred and see if you can detect some changes in the conduction or something like that. It's highly risky and exploratory, but I think it's very exciting that we would potentially have an output for these changes. So basically the future plans then are to looking at how the axon oligodendrocyte unit is formed and maintained and potentially degenerates in disease, and then also how it changes, how all these changes influence axon function. So with that, I would like to acknowledge Chris and their lab. Jaime has been a great mentor, and I've learned a lot thinking about the brain as a bigger picture, multiple cell types working together as opposed to just thinking about myelin and NG2 glia. And also Aaron, we worked a lot on the development of the score imaging. And so thank you everyone for coming, and I'll take questions.
So I'm curious about these long unmyelinated stretches on the mature